Hi class, now we are on Unit 4 and it's all about nucleic acids. This is the part where learning biochemistry becomes exciting because we are getting the answer to the question we've been asking ourselves, like a mystery unlocked. Why do I look like this? Why is my eye color black? Why is my hair curly? Why is my nose shaped like this? And it's all about our DNA, which contains a unique genetic code and is made up of chemical bases in a particular sequence. So the first part is easy to grasp since it is relevant that DNA determines things such as your eye color, your hair color, your height, and even the shape of your nose. But the latter part is where you will start to struggle because it is immaterial, especially when the structure and bonding of the bases are explained. So before we jump to the structure and bondings of the nucleic acids, I would like to introduce to you the amazing people behind the discovery of this to help us examine life at the molecular level. To start with, we have Friedrich Miescher in 1869. So he experimented and isolated a new molecule he termed nuclein, which is now called our nucleic acid. And he isolated this from other cells and later used salmon sperm as a source. Shortly after, Martha Chase and Alfred Hershey were able to show using a bacteriophage that DNA and not protein is the hereditary molecule. One major discovery was by Wilkins and Rosalind Franklin. They studied the structure of DNA crystals using X-rays and they found that the crystals contain repeating subunits, these crystals. Another major discovery is by Erwin Chargaff. He discovered the base pairing rule is between adenine and thymine and guanine and cytosine because of the relative percent of each base. He also discovered that the bonds between each other are by hydrogen bonding. Then in 1953, Watson and Crick used Chargaff's base data and Franklin's X-ray diffraction data to construct the model of DNA that they called the double helix DNA. So the model showed that DNA is a double helix with sugar phosphate backbone on the outside and the paired nucleotide bases on the inside. And the structure fit the spacing estimates from the X-ray diffraction data of Wilkins and Rosalind Franklin, and Chargaff's rule showed that the base pairing was complementary base pairing of a purine and pyrimidine, giving the correct width for the helix. So Watson and Crick not only proposed the double helical structure of DNA and used Wilkins and Franklin's and Chargaff's data, but they also suggested the term central dogma of molecular genetics, which states that genetic information flows from DNA to RNA to protein. So in this unit, we will look at the structure of a nucleotide and differentiate this from a nucleoside and then explain how these nucleotides are linked together to form the nucleic acids. On the next video, we will look at differences between DNA and RNA in terms of structure and function. And on the last video, we will illustrate the biological functions of these nucleic acids in relation to replication, transcription, and translation. 4.1 Nucleosides and Nucleotides. So we start with what are nucleic acids? So nucleic acids are composed of the elements CHON-P or carbon, hydrogen, oxygen, nitrogen, and phosphorus as discovered by Friedrich Miescher. And there are two types of nucleic acids. We have DNA or deoxyribonucleic acid and RNA or ribonucleic acids. These are made up of nucleotides. And nucleotides are made up of three parts. We have our nitrogenous bases, our sugars or the sweet side, and the phosphate group, which is the sour side. For the first part, our nitrogenous bases, we have five. We have adenine, which can be found in both DNA and RNA, thymine, which can only be found on DNA, cytosine, which can be found on both DNA and RNA, guanine, which can be found in both DNA and RNA, and uracil, which can only be found in RNA. So in your book, you can see that nitrogenous bases are divided into two groups. We have purines and pyrimidines. Purines have double ring, while pyrimidines have a single ring. To easily memorize this, I usually use the mnemonic PUGA 2, which stands for purine, and purine has guanine and adenine and have two rings. These nitrogenous bases always pair up in the same way. It's always a purine and a pyrimidine and not a purine and pyrimidine. So it's a purine and a pyrimidine so that we can lessen the steric effect or the bulkiness between the bonding of the molecules. So it's always adenine with a thymine or a uracil and a cytosine with guanine. So I don't know what mnemonic will help you with this one, but this is the mnemonic that I use. So I use atu. Atu is dog in our dialect. So this is adenine pairs with thymine or uracil, and atu here sounds like two because there are two hydrogen bonds between adenine and thymine, while between cytosine and guanine we have three hydrogen bonds. So the second part of our nucleotide are sugars, and these are pentoses. And we number these pentoses in primes. So we have here, we start with C1 prime, C2 prime, C3 prime, C4 prime, and C5 prime. These prime numbers are beneficial when you already start growing your nucleotide structure. So there are two types of sugars. We have ribose, which can only be found in our RNA, and deoxyribose, which can be found in DNA. So what's the difference between the two? So you can see at C2 prime here, deoxyribose lacks an oxygen atom, and this makes DNA more stable because it resists hydrolysis. Take note that the hydroxyl group attached at C1 prime is where the nitrogenous bases will attach themselves through a condensation reaction or through a reaction where there is a removal of water. So the third part of our nucleotide is the phosphate group, this group, and this is the sour side of our nucleotide. This phosphate group makes the DNA hydrophilic or water-loving. So therefore, this is the part that positions itself in the aqueous parts of the cell. So your sugar plus your nitrogenous base will form the nucleoside. So this linkage is via a glycosidic bond, which is always at the beta configuration. So in the examples below, you will see that cytosine plus what sugar is this? So we look at C2, we have an OH, this is a ribose, forms cytidine. Then uracil with a ribose will form uridine. Adenine plus a ribose will form adenosine. Guanine plus a ribose will form guanosine. So remember, your sugar plus your nitrogenous base forms your nucleoside through a glycosidic bond. So nucleoside plus your phosphate group is called your nucleotide. So as you can see here in the example below, we have adenine here plus your sugar. So you can see here that the sugar at C2 doesn't have an oxygen. So this is a deoxyribose. So this as a nucleoside is called a deoxyadenosine. If you remember in our previous slide, if this is a ribose, this is called an adenosine. Okay, so we just add the prefix deoxy if it is a deoxyribose. So with the phosphate group, this is already called deoxyadenosine 5 monophosphate. So the 5' here is because the phosphate group is attached to the 5' carbon of the sugar group, and monophosphate because we have only one phosphate group. So from your activity, you were tasked to write the structural formula of GAC. So let us show how this is drawn. So we start with what is GAC. So GAC is your guanine, adenine, and cytosine. So if you remember our mnemonic, who got 2, guanine here and adenine have 2 rings. The first reaction is the formation of nucleoside, which is joining the nitrogen base and a 5-carbon sugar. The nitrogen from the nitrogenous base will attach to the C1 of the sugar. So it will attach at C1. So we will remove oxygen here. C1. Oxygen will be, water will be removed. C1. If you go back to the concepts of Unit 1, this is a condensation reaction. The second reaction is a combination of phosphoric acid with a nucleoside to form a nucleotide. This is a condensation reaction again between the phosphoric acid and the alcohol group on carbon number 5. Finally, nucleic acids are formed by joining these nucleotides using the same condensation reaction. So you have here the first nucleotide. We have a guanine here plus a ribose. So we term that guanosine. And then we have the phosphate group at the 5'. So that is guanosine 5, 1 phosphate group monophosphate, or GMP. So we will attach this GMP to your AMP and to your CMP to make your nucleic acid. So we follow here the 5' to 3' end direction. So this is 5' to 3', 5' to 3', 5' to 3'. So we remove the oxygen here again. So the nucleotides are covalently linked through the 5' phosphate group to the 3' hydroxyl group of the next nucleotide, creating a phosphodiester linkage. It's called the phosphodiester linkage because you have the phosphate group here and you form two esters, one, two. That's why it's called a phospho, one, two, that's diester linkage, phosphodiester linkage. So for your activity 4.1.1, you practice drawing the structure of all five bases, including numbering of the atoms of the rings, and identify which bases are found in DNA and which bases are found in RNA. For your activity 4.1.2, write the structural formula of DATP, DTTP, DCTP, DGTP, UTP, and give the complete name for each. So you can add additional sheets of paper or bond paper for this activity. So in this topic, we learned how to draw the structure of nucleotide and we differentiated that from nucleoside, and we also learned how the nucleotides are linked together to form our nucleic acid.